Good morning, students. In this session, I am going to discuss about a few more cell organelles. So here we are going to learn what are the main difference between chromosomes and chromatin fibers. Okay, as you learn, chromatin fibers, it's like thin fiber-like structures. They coil together, and what they will form? They will form the chromosomes, right? But we'll see what is the main difference between these two. So first one is chromosome. So you can see here, these are the chromosomes. You can see it's little thick and double. It's like a pair-like. You can see. But here you can see very thin fibers. These are the chromatin fibers. Okay, so this chromosome, this is chromatin fibers. One more thing, you can see this chromatin fibers you can see in the non-dividing cell. This you can see in the dividing cell. Clear? So first difference, chromosome, tightly packed DNA, here unwound DNA. So that is one main difference here. Tightly packed DNA, here unwound DNA. Second difference, found only during cell division. This we can see only during cell division. During the time of cell division, chromatin fibers coil together forming chromosomes, right? So here found only during cell division. Here found throughout interphase. This we can see throughout interphase. The third difference, DNA is not being used for macromolecule synthesis. DNA is not being used for macromolecule synthesis, but here DNA is used for macromolecule synthesis. So these are the three main differences. Okay. Then you can see here, so this is one cell, this is chromosome, this is DNA, double helical structure of DNA. What is a gene? So this chromosome which contains gene. Okay, gene is a basic physical and functional unit of heredity. Genes, which are made up of DNA, act as instruction to make molecules called proteins. So what is a gene? Gene is the basic physical and functional unit of heredity. Genes, which are made up of DNA, act as instructions to make molecules called proteins. So that is a function of gene. The next one is, you can see here some chromosome numbers. So they're given some common names. Human means Homo sapiens. So that is a scientific name. There is a diploid number, 46. Chromosome number is 46. It means 23 pairs. Same like if you are taking monkey, you can see 42. Okay, or 21 pairs we will tell. Same like a dog, 78 chromosomes. Like the few examples they're given here. Then this chromosome, it consists of one chromatid. So this is chromosome consists of one chromatid. This is one chromatid. The center, this is called a centromere. It will duplicate. After duplication, it will become double. Okay, you can see there is one pair. So each one we can call sister chromatid. So this is the centromere. The center, it is called a centromere. It divides into two. So this is one sister chromatid. This is another sister chromatids. Clear? Then this is duplicated chromosome. Then this is centromere. Then after this, what will happen? Mitosis, division. After the mitosis division, you can see here, there are two daughter chromosomes here. Okay, so before first it was one, duplication, it became two. I told you there is a centromere, here it divide, it will become two daughter chromosomes. You can see this is one daughter chromosome, this is second daughter chromosomes. Then what is meant by DNA fingerprinting? Like the fingerprints, the DNA pattern helps in ascertaining the identity of a person, and hence the term DNA fingerprinting. The technique can even testify the percentage of an individual. In a woman's murder case in Delhi in July 1995, the DNA from her unidentifiable cared dead body was matched with the DNA from the body cells of her parents to confirm that they really were the father and mother of the murdered woman. That was one of the earliest cases. Now DNA fingerprinting has become very common. So that is the use of DNA fingerprinting. Then what are the differences between prokaryotic and eukaryotic cells? Prokaryotic means, karyon means nucleus, prokaryon, it means false nucleus. Eukaryon, It means a true nucleus. Here nucleus is with the proper nuclear membrane and all organelles. Here it is without nuclear membrane. So what about the DNA? Here in prokaryotes, DNA is naked, DNA is circular, usually no introns. In eukaryotes, DNA bound to protein, DNA is linear, usually has introns. Clear? So here DNA is naked, DNA bound to protein. DNA is circular, DNA is linear. Usually no introns, usually has introns. Then what are the difference in organelles? Here no nucleus, has a nucleus. No membrane bound, here nuclear membrane is present, membrane bound. Then 70S ribosomes, here 80 ribosomes. Then what about the reproduction? Prokaryotes, you can see binary fission. Here mitosis and meiosis. Then second difference in the reproduction, here single chromosome, haploid. Here chromosomes paired, diploid or more. Then what is the average size? Smaller, 1 to 5 micrometer. Here larger, 10 to 100 micrometer. Then what is the, in the figure you can see what is the difference between eukaryotic cell and prokaryotic cell. So this is a eukaryotic cell. You can see the nucleolus inside the nucleus. We told there is a nucleolus present. Then membrane enclosed nucleus. So the nucleus is with a nuclear membrane. Then mitochondria, this is mitochondria. Then the ribosomes are present. So that is eukaryotic cell. Then cell membrane. This is cell membrane or plasma membrane. So this is true nucleus. Eukaryotic means animals or organisms will have a proper nucleus or true nucleus. Here prokaryotic means false nucleus. Okay. Here you can see these are the small ribosomes. This is nucleoid. You can see there is no proper nuclear membrane. This is nucleoid. Then cell membrane. Here you can see three membranes. First black color, that is cell membrane. Second layer blue color, that is cell wall. Then third, you can see in some prokaryotes, you can see capsule. See third layer. So there are three layers. First layer, second layer, third layer. First layer cell membrane, second layer cell wall in some eukaryotes. Then, sorry, in some prokaryotes. Then third layer, there is capsule, there is also in some prokaryotes. This is flagella. Cell organelles. So this is cell membrane, cytoplasm, nucleus. These are the cell organelles. Yes. The next is mitochondria and structure. Mitochondria, actually it has two membrane, outer membrane and inner membrane. So what is outer membrane? It is smooth and is composed of equal amount of phospholipids and protein. It's very smooth in nature. Outer membrane is very smooth. So you can see two layers. Outer layer is very smooth and is made up of phospholipids and proteins. It has large number of special protein. They are known as porins. So it has large number of special type of protein. They are known as porins. The porins are integral membrane proteins and they allow the movement of molecules that are of 5000 Daltons or less in weight to pass through it. So they are the special type protein. They are known as porins. What is function of the porins? They are the integral membrane proteins and they allow the movement of particles or molecules that are of 5000 Dalton or less in weight to pass through it. The outer membrane is freely permeable or it will allow any molecule to go inside or go outside. Okay, that is called freely permeable to nutrient molecules like to nutrient molecules, ions, energy molecules like ATP, ADP, etc. So they are freely permeable to nutrients, ions, energy molecules like ATP, ADP, etc. Then what about the second, inner membrane? What is the specialty of inner membrane? The inner membrane of mitochondria is more complex. It is folded into a number of folds many times. It is known as cristae. So inner membrane, little complex type, it is making many foldings, that is known as cristae. These foldings help to increase the surface area inside the organelle. So why it is making like many foldings? It increases the surface area inside the organelle. The cristae and the proteins of the inner membrane aids in the production of ATP molecules. So what is the function of this cristae? This foldings, cristae, and the proteins of the inner membrane. It helps in the production of ATP molecules. Okay, it helps in the production of ATP molecules. Various chemical reactions takes place in the inner membrane of the mitochondria. Same like many chemical reactions takes place in the inner membrane of mitochondria. Clear. So inner membrane, one function is production of ATP molecule. Second function, many chemical reactions taking place in the inner membrane. Unlike the outer membrane, the inner membrane is strictly permeable. It will allow only few things. It is permeable only to oxygen, ATP, and it also helps in regulating transfer of metabolites across the membrane. So it is very selectively, it will not allow all the things to get inside or go outside. It is very selectively, strictly permeable. It is permeable only to oxygen and ATP. And it also helps in regulating transfer of metabolites across the membrane. Then what is the structure of mitochondria? Just now I told the same thing. Then in between, you can see one more thing here. That is, yeah. First I will show you. So this is outer membrane. We told it's very thin membrane, outer membrane. Then inner membrane, I told it's little complex type. There you can see the foldings. This folding is called cristae. Clear. So first one is here. This is a purple color. This is DNA. The round structures, they are ribosomes. Then here little jelly-like fluid is called matrix. And this is outer membrane. This is inner membrane. Intermembrane space. Okay. Between outer and inner, you can see one space that is called intermembrane space. Cristae junction. So this folding is called cristae. This is a junction, cristae junction. Then F0 and F1 complexes that you can see here in this space inside the cristae. You can see F0, F1 complexes. So this is a structure of mitochondria. Okay. So the next, this intermembrane space and matrix. Intermembrane space means, just now I showed you, it is a space between outer and inner membrane of the mitochondria. It has the same composition as that of the cell cytoplasm. So this space, okay, this is known as intermembrane space. It has the same composition as that of the cell's cytoplasm. There is a difference in the protein content in the intermembrane space. Only the difference in the case of protein content in the intermembrane space. Second, the last one is matrix. Yeah, this one here, this is a matrix. The matrix of the mitochondria is a complex mixture of protein and enzymes. Matrix means it is a mixture of proteins and enzymes. These enzymes are important for the synthesis of ATP molecules, mitochondrial ribosomes, tRNA, and mitochondrial DNA. Okay, so this part, this is matrix. It is a mixture of proteins and enzymes. So what is the function of these enzymes? These enzymes, the important function of the enzymes are for the synthesis of ATP molecules, mitochondrial ribosomes, tRNAs, and mitochondrial DNAs. Okay. Thank you, students.